today we'll see some important crude drugs and introduction to glycosides. So this part is also again important for your exam. So we'll see first of all what do you mean by glycosides? Okay, it is made up of two terms, glyco and sides. Glyco means sugar and sides means side chain. So you can correlate this. Glycosides means it is made up of two components. First one is a sugar portion and another one is a non-sugar portion. Now this sugar portion is known as a glycone, non-sugar portion is known as a glycone, gene. Okay, so glycosides are nothing but the organic compound from plant and animal sources which on enzymatic hydrolysis or acid hydrolysis will give you two components. First component is known as sugar and second component is known as non-sugar. Sugar part is sugar portion is known as a glycone and non-sugar portion is known as a glycone or genin. Now chemically they are nothing but the acetals of sugar or ether. Esters of sugar and ether. They are formed by interaction between hydroxyl group of non-sugar part and sugar moiety. So two hydroxyl group they will come together to form a bond with the loss of water molecule. That is nothing but the glycosidic bond. So bond involved is nothing but the glycosidic bond. Now in this bond there might be a different atoms like carbon, oxygen, sulfur, nitrogen, etc. etc. So depending on that also they are classified in different ways. So again chemically they are the acetals of sugar. They are formed by interaction of hydroxyl hydroxyl group of non-sugar and sugar with the loss of water molecule. The hydroxyl group of a glycone may be either alcoholic hydroxyl group or phenolic hydroxyl group, or in some cases it might be a aminos. Most commonly found sugar in the glycoside, that is a beta D glucose. Remember, most commonly sugar कौन सा होता है? Beta D glucose. Other sugars are also found, but they are less abundant. So most commonly found sugar is a beta D glucose. Remember, most commonly found sugar is a beta D glucose. Other sugars like galactose, rhamnose, mannose, digitoxose, that is a special one in digitalis. So these kind of sugar also found in the glycosides. So non-sugar part that comes from the different metabolic pathway in the plants like cyclic acid pathway or mevalonic acid pathway. So non-sugar part may be either it may be either derived from amino acid or derived from your nucleosides. So that depends. But that non-sugar part plus sugar part they are joined together by glycosidic bond, and that is nothing but the glycosides. Okay. So plant mostly consists of beta glycosides. प्लांट में जो है वो बीटा ग्लाइकोसाइड्स ही होते हैं मैक्सिमम, बट थेरेटिकली बोथ ग्लाइकोसाइड्स आर पॉसिबल, अल्फा प्लस बीटा, बट मैक्सिमम जो प्लांट में होते हैं दैट इज अ बीटा ग्लाइकोसाइड्स. So beta glycosides are plant consist of only beta glycosides. Now you can see over here it is a structure of one glycoside. So this part is sugar portion. This part is the sugar portion known as a glycone part, okay, or sugar part, and this part is known as a glycone part. This part is known as a glycone. Okay, a glycone part or genin or a glycone part. Now this a glycone part, this comes from different metabolic pathway. This comes from a different metabolic pathway, and this sugar and a glycone part they are joined together by glycosidic bond. They are joined together by this glycosidic. During the formation of glycosidic bond, so oxygen may be there. There may be a carbon. There may be a sulfur atom. There may be a nitrogen atom. So depending on that, different glycosides are classified. Like C glycosides, O glycoside, N glycoside, S glycosides. Okay, and depending upon the nature of a glycone part, depending upon the nature of a glycone part, so they are also classified depending upon the nature or structure of ring present. So for example, in this example, the ring is nothing but the anthraquinone. Okay, that's why it is an example of anthraquinone glycoside. Okay, so remember over here this most common sugar found in the glycoside, that is nothing but the beta D glucose, and only and plant contains only beta glycosides. So we'll see the next point. So we'll see the classification of these glycosides. Classification of these glycosides again. So उसके ऊपर GPAT में क्वेश्चन आता है. So depending upon the nature of a glycone part, depending upon the nature of a glycone part, glycosides are classified in different ways. So first one is known as anthraquinone glycoside. Anthraquinone glycoside or anthracene. Okay, so here anthracene ring will be present either anthracene single ring or with the ketone, that is anthraquinone. Okay, so whatever the glycosides present in the Senna, rhubarb, then your Senna, rhubarb, aloe vera. So all these glycosides, they are nothing but the anthraquinone glycoside like barbelloin, then globulin, then senosides, etc. etc. Then second class is nothing but the sterol glycosides. Sterol, or they are also known as a cardiac glycosides. Sterol or cardiac glycosides. So here the nature of a glycone part will be steroid in nature. Steroidal a glycone part will be there like cyclopentanone or hydropenanthrin. Okay. So in this case, for example, digital is thioetine, squil, etc. etc. That will have steroidal a glycone. So that's why steroid glycoside. Then third one is saponin glycosides. So they will have a soap like action. So आपका जो soap होता है, so soap का क्या होता है, foam होता है. Okay, foam बनता है soap से. So if saponin glycoside, they are dissolved in water, and if they are shaken in a test tube, they will produce a foam. Okay, they will produce a foam, and they have a hemolytic activity also. So saponin glycosides. So here also we have different a glycone parts. For example is diosine. Okay. So then next one is a cyanogenetic glycosides, or they are also known as cyanophoric glycosides. Cyanogenetic or cyanophoric glycosides. For example, bitter almond, wild cherry bark. Bitter almond, wild cherry bark consist of a cyanogenetic or cyanophoric glycoside. Now इसके ऊपर एग्जांपल आ सकता है आपका. Now next is a isothiocyanate glycosides. Isothiocyanate glycosides. Okay. So for example, cinegreen and cinnabalin. Cinegreen from black mustard and cinnabalin. So these are the isothiocyanate glycoside. So this was a question again in a GPAT. Okay. So important cyanogenetic glycosides, isothiocyanate glycoside. Isothiocyanate particularly found in the cinegreen from black mustard. Okay. So इसके ऊपर क्वेश्चन आ चुका है. Next one is a flavonoids or flavonol glycosides. So they are present in the plant like ginkgo biloba or rutin or ginkgo biloba, ruta plant or ginkgo biloba plant. So rutin or ginkgolides, is that is nothing but the flavonoid glycosides. Now again see, next one is a coumarin or furanocoumarin glycosides. So here they will have a hydroxychromone or coumarin ring in the structure. So for example, this hydroxycoumarin glycoside. Next is aldehyde glycosides. Aldehyde glycosides. So example is vanillin. Vanillin consists of aldehyde glycoside. Vanillin consists of aldehyde glycoside. Okay. So this was again a question in GPAT or various kind of exam. Important. Aldehyde याद रखो और isothiocyanate, cyanogenetic glycoside, flavonoid वगैरह याद रखो. Then next one is your phenol glycoside or alcohol glycosides. So it is present in the salicin from onion. Then steroidal glycoside again. For example, diosgenin. Sterol and steroidal is little bit difference. In case of sterol, you will have same ring but additional hydroxyl group. Here you will have not hydroxyl group. Next one is known as a bitter glycosides. Next one is known as a bitter glycosides or glycosidal bitter. For example, in case of gentian, then picoryza and chirata. These are the examples of glycoside depending on the nature of a glycone part. So depending upon the nature of a glycone part, they are classified into anthraquinone, sterol, saponin, then isothiocyanate and flavonoid. Okay, so remember these things. This is important. Okay, coumarin, furanocoumarin, aldehyde glycoside होता है vanillin में, आपका जो vanilla फ्लेवर होता है. Okay, then isothiocyanate cinegreen में होता है. cyanogenetic में जो bitter almond होता है, wild cherry bark होता है उसमें होता है. Then steroidal glycoside इसमें होता है, diosgenin में होता है. Now depending upon the nature of linkage, depending upon the atom involved in the bond formation, they are classified in different way. For example, C glycosides. So here carbon will be involved in the carbon atom will be involved in the formation of glycoside bond. So anthraquinone glycosides such as, for example, such as cascarosides from Cascara, C glycoside. They are known as C glycoside. So cascarosides from Cascara bark, then aloe from aloe vera. There are some members of flavon type of glycosides also, which are a C type of glycoside. Next one is O type of glycosides. So here oxygen will be will be the atom that is involved in the glycosidic bond. So very common in the plant like Senna, rhubarb, and Pranula. Pranula, Senna, rhubarb. Then next one is yes glycosides. So yes means sulfur containing glycosides. Okay. So isothiocyanate glycosides, particularly cyclin from black mustard. We have seen this cyclin from black mustard. Then nucleosides, N glycosides or nucleosides. So N glycosides they are present. For example, or whatever the nucleotide bases are there. So adenine, guanine, purine, pyrimidine. So they used to form a bond with the sugar, and they are known as N glycosides. Okay. So this is again just classification depending upon the type of bond present in the glycosides. So type of atom present in the glycoside bond. Okay. So for example, this is the structure of your aglycone part or gelin part. So depending upon the nature of that, they are classified as. So first we will see the anthraquinone glycosides. You can see here the ring is present, that is known as anthracene. So three rings joined together, it is known as anthracene, and there is a diacroton over ring B. So that's why anthraquinone. Okay. So then it is just a modification of that. So oxyanthrone. So that is hydrolyzed containing. Then anthranol means total alcohol. So anthanol. So all the anthraquinone glycosides, they are their side chain is from this structure only. Okay. So dianthrone means, so two anthrone they are joined together by bond between 10 and 10 dash. 10 dash bond. That is a dianthrone. And if alcohol is that dianthranol. So for example, this is a aloe. Is also anthracene. So now there is a particular test for identification of anthraquinone glycosides. So that is nothing but the there are two tests for O glycoside. There is one test known as a Brown Tracker test, particularly for O glycoside, and this modified Brown Tracker is for particularly for C type of glycoside. Remember these two things. For O glycoside, you have modified. Sorry, you have Brown Tracker test. For modified for C type of glycoside, you have modified Brown Tracker test. So what is in this? So whatever glycoside sample we have, that is powdered and hydrolyzed with the first. In case of Brown Tracker test, the sample is hydrolyzed by sulfuric acid. Okay, it is hydrolyzed by sulfuric acid. Then you have to filter it. Filter it plus you have to add a organic solvent like ether, benzene or ether, and benzene, ether or chloroform. And then you have to separate the organic layer. Okay, you have to separate the organic and aqueous layer by separating panel, and then you have to add ammonia. Now ammonical layer will show pink color due to presence of anthraquinone glycosides. That is nothing but the Braun Trager test, and that is specific test for O type of glycoside. O glycoside Braun Trager. Now modified Braun Trager just replace this hydrochloric replace this sulfuric acid with hydrochloric acid. Okay, and then add perichloride. So powder drug plus hydrochloric acid, perichloride. And then now boil it, extract. Okay, boil it and then add your organic solvent like carbon tetrachloride or benzene. Then again same procedure, filter it and add ammonia. Ammonical layer will show again rose red color. Ammonical layer will show pink or rose red color. So that indicate the presence of anthraquinone glycoside, particularly C type of glycoside. Particularly C type of glycoside. इसके ऊपर क्वेश्चन आता है. So Brown Tracker test is for O glycoside. Modified Brown Tracker is for C type of glycosides. All the anthraquinone glycosides they will consist of this structure. You will have such kind of structure in life. Dianthrone, dianthranol. Then you this okay, it is a glycoside present in the aloe vera. This Braun Tracker test and modified Braun Tracker test. So Braun Tracker test is for O glycosides and modified Braun Tracker test is for C glycosides. Okay, so that will again एक बार आने वाला है वो टेस्ट. So now first drug from anthraquinone glycoside. So anthraquinone glycoside का पहला ड्रग आता है आपका प्रोडक्ट, that is known as Senna. Senna is also known as Indian Senna. Then also known as Senna leaf. Then also known as Senna की पत्ती. Also it is known as Tinivali Senna or Cassia Senna. So all these are the synonyms for Senna. Now what is the biological source? So it is nothing but the dried leaflets of Cassia angustifolia. Cassia angustifolia, family Leguminaceae. Cassia angustifolia, family Leguminaceae, and it should contain not less than 2% of anthracene derivative or glycoside calculated as सेनोसाइड सेनोसाइड बी होना चाहिए ठीक है सेनोसाइड बी not less than 2% of senoside B. Now it is cultivated in the various part of India. It is mainly done in the south region. Okay, mainly done in the Tinivali. Tinivali is a district. Okay, then Madurai is also district. Ramanathapuram district. So it is cultivated in the south, particularly in the Tinivali, Madurai, Ramanathapuram district of Tamil Nadu, Kadapa district of Andhra Pradesh, and some part of Kutch in the Gujarat. That is the geographical source now. Cultivation and collection. Now there was a question in GPAT 2019 on the cultivation collection of this Senna. So cultivation collection के ऊपर 2019 में क्वेश्चन आया था. So for to for the cultivation of this Senna, you need red loamy or Chacos gravely soil. Red loamy मतलब लाल माटी, Chacos gravely मतलब वो थोड़ा सा खुरदुरा soil चाहिए उसको. Red loamy Chacos gravely soil is required, but it can grow also in the alluvial loamy. It can grow also in alluvial loamy soil. So that is the type of soil. Then well plugged, harrowed, leveled land is required. Means whatever field where you want to grow, where you want to cultivate this Antharctic Senna. So there you have to well plugged land, harrowed and leveled. उसको नांगरनी वगैरह करना, उसको लेवल करना. Sowing is done in the sowing of this Senna seed is done by broadcasting method. Remember broadcasting method. Simple है, सिर्फ इंग्लिश में थोड़ा सा है. So it is done by broadcasting. Broadcasting method means शिंप नहीं, ठीक है, मतलब बिया शिंप नहीं. So that is the broadcasting method. It is done in the February or March. It is done in the February or March. So first sowing is done in the February to March, and second sowing is done in the October November. पहली sowing में अगर crop नहीं raise होता है, seedlings are not there, then you have to do second sowing. And surface of the seeds they are abrased. Surface of seeds they are abrased with the sand by triturating with the sand. Surface of seeds are abrased by triturating with the sand. It is a semi-irrigated plant. Remember semi-irrigated plant. इसको ज़्यादा वाटर नहीं चाहिए, तो it is a semi-irrigated plant. After the growth of 3 months of sowing, the leaves are harvested when the leaflets are thick, fully grown and greenish in the color. 3 महीने के बाद उसको हार्वेस्ट कर सकते हो आप, उसका लीव्स वगैरह लीफलेट निकाल सकते हो जब लीफलेट जो है ग्रीन कलर हो जाएंगे. Now senoside content is maximum in the leaves. Senoside contents are maximum in the leaves which are fully grown and goes decreasing on the maturity of. जैसे जैसे वो मैच्योर हो जाएगा, मतलब बड़ा हो जाएगा, तो उसका सेंसर कंटेंट डिक्रीज होता है. आफ्टर द कलेक्शन द लिपलेट्स आर ड्राइड इन द इंडोर. घर में ही उसको स्प्रेड करके ड्राई करना. स्प्रेड इनटू थिन लेयर ऑन द फ्लोर एंड ड्राई. देन पैक्ड इन द बेल्स अंडर हाइड्रोलिक प्रेशर. बड़े बड़े जो ड्रम्स होते हैं उसमें पैक करना है उसको hydraulic pressure. That is nothing but the cultivation collection. In GPAT 2019, the question was like that: short-term drought. What is the effect of short-term drought on the senoside content in the senna? The question was like that. What is the effect of short-term drought? And the option was like increase in the senoside A and B content. Option was like that: decrease in the senoside A and B content, increase in senoside A and decrease in senoside B content, likewise that. Okay, so remember short-term drought increases the senoside content in the plant. Okay, उसका जो सेनोसाइड्स होता है, सेनोसाइड मतलब ग्लाइकोसाइड होता है उसमें. ओके, सेनोसाइड शॉर्ट टर्म ड्रॉट. व्हाट इज मीन बाय ड्रॉट? ड्रॉट मीन शॉर्टेज ऑफ वाटर, या फिर आप बोल सकते हैं ड्रॉट मीन्स दुष्काल. तो कुछ समय के लिए उसको पानी बंद करना हो प्लांट को, बोलते हैं ड्रॉट. So short term drought increases the senoside content in the plant because of it will mature early. So short term drought result in the increase in the senoside content in the plant. Remember this. This was question in GPAT 2019 as it is a semi-irrigated plant. We'll see the next point, that is chemical constituent and microscopy and all that. So microscopic character. So isobilateral leaf is there. Okay, so in case of Senna, isobilateral leaf is there. Isobilateral leaf. Then epidermis with unicellular conical thick and warty trichomes. यहाँ पे देखो ये क्या है, isobilateral leaf है, trichomes है उसमें, they are present on the both surface because the leaf is isobilateral. Now rubiaceous or parasitic stomata, cluster of calcium oxalate in the parasite tissue, okay, which is present on both sides. Then step rectangular cell, spongy parenchyma. These are the microscopic characters. Patch of sclerenchyma towards the upper epidermis, about the xylem is present. Presence of cluster sheath of cholenchyma is a characteristic of Senna leaves. Non-lignified unicellular covering trichome. So just remember, rubiaceous or parasitic stomata, cluster of calcium oxalate, then presence of cluster sheath of cholenchyma. That is the microscopic characteristics of your Senna leaves. Non-lignified covering. So that is the microscopic character. So chemical constant इसमें क्या है? So obviously it will have anthraquinone glycoside known as sinusoid A and sinusoid B. Two anthraquinone glycosides are present in the Senna, sinusoid A and sinusoid B. So out of which sinusoid B is responsible for the purgative activity. ठीक है, यहाँ पे देखो sinusoid B is responsible for the purgative activity. इसके ऊपर भी क्वेश्चन आ सकता है. So out of these sinusoid A and sinusoid B, which is responsible for the purgative activity? So sinusoid B is responsible. So sinusoid A and sinusoid B, they are nothing but the stereoisomers of each other. They are dimeric glycoside with rein dianthrone as a glycol, and 10-10 bis or 10-10 di 9-10 dihydro 1 dihydro 9 oxan oxanthrazine oxanthrazine 3 carboxylic acid, उसका एक हाइपेक नेम है. So that is हाइपेक नेम of that. So we'll see again. So 10 dash bis 9-10 dihydro 1,8 dihydroxy 9 oxanthrazine 3 carboxylic acid. That is a IUPAC name of that. Okay. Remember over here again, A glycone part of sinusoid A is a dextrorotary and sinusoid A is a dextro, sinusoid B is a meso. Sinusoid B का जो A glycone part है, that is a mesoform. Sinusoid A का जो है, that is dextro. Other constant like sinusoid other constant like sinusoid C and sinusoid D are also there. They are nothing but the heterodianthrone. Rein, aloe, rein, 8-glucoside, camphorol are the other constants. Phytosterol, mucilage, resin, myrcene, alcohol, and salicylic acid is also present. Phytosterol, mucilage, resins, myrcene, alcohol, and salicylic acid. But what is important over here? Sinusoid A and sinusoid B. So out of that sinusoid B जो है आपके negative activity दिखाता है sinusoid B. And out of this sinusoid B is a meso. We'll see it again. I will demonstrate these things with structure also. We'll see the uses of this. Now this Senna is used as a laxative and purgative. Senna is used as a laxative and purgative. So it is used in the habitual constipation. See uses. What are the use of this Senna? It is nothing but it is used as a purgative in a habitual constipation. Purgative, laxative. There are two terms, purgative and laxative. So laxative means any drug uh, that can increase the stool formation, but the stool formed they are smooth and non-irritating, but they are solid. If stools formed are solid and smooth, that is known as a laxative. If any agent is able to increase your stool formation, but stools are not solid, they are liquid, then that is known as a purgative. Okay, and there is one more term known as saline cathartic. There are three terms related to this. These are three terms: laxative, purgative, and saline cathartics. So laxative means any agent that can increase the stool formation. Consistency of stool is solid. That is nothing but the laxative. Now, what is purgative? Purgative means any agent that can increase the stool formation. The consistency of stool is like a liquid. Okay, liquid stool हो जाएगा diarrhea type. Nothing but the purgative. What is meant by saline cathartic? Saline cathartics are these are the agent which will increase the stool formation. The stools will be like a watery, ice water diarrhea type के stool. Total loss. ठीक है? So that is nothing but the saline cathartic. So laxative, purgative, and saline cathartic. These are the three terms. Okay. So this Senna is used as a purgative in habitual constipation. Now this Senna first they are absorbed from the intestine in the blood. They are hydrolyzed and separated. The glycone part is again excreted in the colon. क्या होगा इसका GIT में से एब्सॉर्प्शन होगा, ब्लड में इसका हाइड्रोलाइसिस होगा और जो ए ग्लाइकन पार्ट है वो एक्सक्रीट होगा कहाँ पे, इंटेस्टाइन में ही री एक्सक्रीट होगा इंटेस्टाइन में. The A glycan part will irritate the intestine and stimulate the colon for removal of stools form. That is a purgative action. Now griping effect is due to the resin griping effect of this senna. Griping effect means, so griping effect means ज़्यादा purgation purgative effect मिलेगा, totally आपको बाथरूम में ही बैठना पड़ेगा. So griping effect means that stoppage of that. So griping effect is due to resin content that is found in the senna, that is imodine, resin or imodine content. उसका chemical test we have seen, that is nothing but the brown tracker test. You have to take a drug, hydrolyze with HCl, then add water immiscible solvent or organic solvent like ether benzene or chloroform, soda or ammonia. Okay, so ammoniacal layer will show pink color. That is nothing but the presence of anthraquinone glycoside. Now this test is negative in case of anthraquinones. एंथ्राक्विनोन है वो पॉजिटिव देगा, एंथ्रानॉल से वो नहीं देगा. नहीं, एंथ्रानॉल्स आर नथिंग बट द reduced form of anthraquinone. Reduced form of anthraquinone, that's why anthranol's will not give this positive. There is another test for detection of this antranol. Anthranol's are detected by fluorescence test, which show strong fluorescence in alkali, but anthran will not show any fluorescence. So adulteration and substitution. So this is again an important part for Senna. Adulteration and substitution. Senna is adulterated with a different variety of other Senna variety. So what are they? That are nothing but the dog Senna, then Palte Senna, then okay another Bombay Senna and all that, Mecca Senna. We'll see it one by one. So substitution and adulteration. So first Tinivali Senna. Our original drug is nothing but the Tinivali Senna, Cassia angusticuli. Now it is substituted with the different variety of Senna. First one is a dog Senna, known as a Cassia ovovata. Okay, Cassia ovovata is dog Senna. So it is distinguished by ovovate leaves, papillose cells in the lower epidermis and contain only 1% glycoside. So whereas your original drug consists of 2% glycoside, that is Senna B. Senna कहाँ से मिलता है? Cassia ovovata. Okay, remember. नाइपर के लिए पूछते हैं ये क्वेश्चंस. Then next one is a Palte Senna, that is known as a Cassia auriculata. So in this variety, you will get a you will not have any anthraquinone glycosides. So absence of anthraquinone glycosides shows crimson color and leaves are treated with the solution of chloral hydrate. ये पालते सेन्ना का अगर leaves अगर ट्रीटमेंट किया chloral hydrate के solution के साथ, तो you will get a crimson color. Okay, other varieties are also like Bombay Senna, Mecca Senna, Arabian Senna. They are also used as a substitute for this original Tinivali Senna. There is another drug known as a Corirea multifolia. Corirea multifolia, which is a highly poisonous drug, and presence of this drug in the Senna should be recognized, because if it is present, it may poison you. Okay, Cassia momenta is another variety known as a false Senna. Cassia momenta जो है वो variety है Senna का, that is used as adulterant. It is nothing but the false Senna. Different varieties of Senna used as a adulterant. कितने variety? Dog Senna, Palte Senna, then Bombay, Mecca, Arabian Senna, then Corirea multifolia, then Cassia momenta, which is known as a false Senna. These are the adulterant for original drug. Now allied drug. So drugs similar to our original drug, which are they are, that is nothing but the Alexandrian Senna. Okay, Cassia Senna जो है ओरिजिनल है, उसके जैसे ही दूसरा है Alexandrian Senna. So the synonym for this is known as a Foley Senna, Cassia Senna, Egyptian Senna. Okay, the biological source is nothing but the Cassia acrotipolia. So originally जो Tinivali Senna है उसको बोलते हैं Cassia angustifolia. So that is a Tinivali Senna. Okay, and Alexandrian Senna is known as a Cassia acrotipolia, family Leguminaceae. Other parameters are similar to the Tinivali Senna, only the leaf constant are different. मतलब पैलेसाइड रेशो दोनों का अलग है, stomatal index दोनों का अलग है. बाकी remember chemical constant, uses and adulterants especially. These are important for your GPAT as well as other exams. We'll see the next drug. See the structure of your sinusoid A and B. Sinusoid A and B. So you can see the structure of sinusoid A and B. We'll explain this. So this is the structure of your sinusoids. Structure of your sinusoid. Okay. So it is nothing but the dianthrone. Okay, dianthrone. Now sinusoid A, sinusoid B, sinusoid C, and sinusoid D. So what are they? So sinusoid A. Sinusoid in the sinusoid A, this R group. This R is nothing but the carboxyl group. Okay, and the configuration at this position, this 10th position, यहाँ पे एक हाइड्रोजन होगा आपका ऐसे यहाँ पे एक हाइड्रोजन. Now because of that, this is a your this is your asymmetric carbon atom, and that's why there is a geometrical isomerism possible over there. So in sinusoid A, COH group is there R, and the configuration at 10 and 10 dash, so that is a trans. Okay, in case of sinusoid B, again R group is a carboxylic acid group, and the configuration at 10 dash is a meso. Okay, sinusoid C में होता है आपका मिथाइल अल्कोहल CH2OH ग्रुप होता है R, एंड कॉन्फ़िगरेशन होता है ट्रांस. सिनोसाइड D में आपका कॉन्फ़िगरेशन होता है मेसो. जस्ट रिमेंबर दिस. योर सेन्ना प्लांट विल लुक लाइक दिस. सेन्ना का प्लांट ऐसा होता है. यू माइट हैव सीन इन द रोड साइड. रोड के साइड पे भी मिलता है ये सेन्ना प्लांट. इट इज यूज्ड एज अ laxative and sedative. It is also cultivated medicinally. The next point. Next drug is known as a rhubarb. Next drug is known as a rhubarb. Okay, the synonyms for this rhubarb is nothing but the radix or rehum or radix rei or revandachini. These are the synonyms. Now, what is the biological source? So it is a dried rhizomes. जो rhizomes होते हैं उसका. Of rehum imodi. Rhizomes of rehum imodi. That is known as the Indian rhubarb. Rehum imodi is known as the Indian rhubarb, and Rehum palmatum. Another variety, Rehum palmatum. Okay, that is Rehum palmatum is another variety. Rehum webianum, that is known as the Chinese rhubarb. Indian rhubarb is nothing but the Rehum imodi, Rehum palmatum. Rehum webianum is nothing but the Chinese rhubarb. Family Polygonaceae. Polygonaceae family का है. So they are found in the southeast China, then Tibet, Korea, Kashmir, Sikkim region of India, West Germany, and European countries. So cultivation collection is not important over here, but you just remember it is a drought resistant plant. It is a drought resistant plant. So if you this plant requires water. Okay, so if there is a drought, so the content of active drug will be decreased. So it is a drought resistant. It is a drought resistant means it can tolerate a drought. अगर सूखा पड़ गया वहाँ पे फिर भी ये जिंदा रहता है प्लांट. ड्रॉट रेजिस्टेंट प्लांट. Rest of the things are not much important. Microscopic characters are also not important for this. Just remember these things. Biological source, the variety. Okay. Which one is a Indian? Which one is a Chinese? So Rehum imodi is nothing but the Indian variety, and Rehum webianum is nothing but the Chinese variety. Remember. Now chemical constant क्या है इसमें? So 2 to 4.6% of total glycosides. So again anthraquinone glycoside with carboxyl group, for example rein and glucurine. Anthraquinone glycoside with that carboxyl group like aloe, chrysophenol and bicycone. इसका स्ट्रक्चर तो बाद में दिखाऊंगा. Then anthraquinone and dianthrone like aloe, imodine, chrysophenol. Heterodianthrone like palmidine A and palmidine B, palmidine C. So these are the three main: palmidine A, palmidine B, palmidine C. So what is palmidine A? Aloe anthrone plus imodine dianthrone. Palmidine B, aloe anthrone plus chrysophenol anthrone. Palmidine C, imodine anthrone plus chrysophenol anthrone. So not important. अपने ये बोल भी गए तो कोई दिक्कत नहीं. So it is not important. Remember, astringent part of this rhubarb consists of gallic acid, glucogalline, tannin, catechin, epicatechin. All these are nothing but the tannin content, tannin or tannic acid content, and because of that, the drug is astringent. So astringency of rhubarb is due to particular tannin known as a ribotanic acid. So astringent content or astringency. So astringency means the protein precipitation. It can precipitate your protein. Okay, it's like amla. So astringency of this rhubarb is due to rheotannic acid. Remember this line. It is used as a bitter stomachic purgative. Stomachic means that will increase the uh, gastric. Okay, so it can increase the your gastric acid secretion or gastric fluid secretion, that is known as a stomachic. Okay, then purgative means I have explained what is purgative. So again, the taste is nothing but the modified prawn tracker taste. Okay, तो सेना का जो है सेना में क्या था प्रॉन ट्रैगर टेस्ट बिकॉज सेना कंसिस्ट ऑफ ओ ग्लाइकोसाइड्स but this rhubarb consists of C glycoside. That's why modified prawn trigger test. So here you have to hydrolyze the drug with hydrochloric acid, then hydrochloric acid and perchloride, then filter, separate the that, and then add your organic solvent like benzene or carbon tetrachloride. Add ammonia solution. Pink color will get. Okay. You have your syllabus have mentioned only few drugs, but I'm taking all because who knows, they can ask anything. So then allied drug. So इसके similar drug कौन से like your original drug. So Indian rhubarb or Himalayan rhubarb or Revant chini. Indian rhubarb is known as a Himalayan rhubarb or Revant chini. Now another drug is known as a Reham Vebianum, known as a Chinese Reham Vebianum, known as Chinese rhubarb. It consists of 1,8 dihydroxy 3 hydroxy methyl 3,5 dihydroxy methoxy stilbene. Due to this stilbene content, so it is estrogenic in nature. Stilbene content. Reham imodi. Reham imodi found in the India and Nepal shows deep violet fluorescence under UV light, and it contains one hydroxyl. It contains one hydrolyzable tannin known as a rapnoticin. It does not contain this hydrolyzable tannin rapnoticin. Then the rapnotic rhubarb is another known as rapnotic rhubarb. So it is obtained from Reham rapnoticum, which consists of rapnoticin, which is a crystalline glycoside, and it is a estrogenic in nature. Estrogenic in nature means so it will increase the, it is agonist to estrogen receptor. तो लेडीज में एस्ट्रोजन का लेवल बढ़ाएगा ये. So the contain 3,3-4 tetrahydroslivin which show blue fluorescent under UV. Its odor is sweet and doesn't contain rein, imodine and aloimodine. आपको इतना याद रखना है कि जो ओरिजिनल रूबाब है, दैट इज इंडियन रूबाब नोन एज हिमालयन रूबाब और रेवंत चीनी. रूबम रेयम वेबियनम जो है दैट इज चाइनीज रूबाब, उसमें स्टिल्बिन होता है, सो दैट इज आल्सो ईस्ट्रोजेनिक इन नेचर. सो दैट इज ओके. सो लेडीज में कॉन्ट्राइंडिकेटर होता है. मॉल रेयम इमोडी तो कोई दिक्कत नहीं. देन रैप्नोटिक रूबाब जो है रेयम रैप्नोटिकम. So it consists of one rapnoticin, that is non-hydrolyzable tannin, okay, and consists of a crystalline glycoside known as a stilbene. So again it is estrogenic in nature, so it is contraindicated in the ladies. Rapnotic rhubarb बारे में नाइपर में क्वेश्चन. These are the A glycon, structure of A glycon. So imodine का स्ट्रक्चर कैसे होगा, अलोइबोडन का कैसे होगा, क्राइसोपेनॉल क्या है, फाइसिकोन क्या है, ठीक है. सो इमोडिन मतलब देखो सिर्फ यहाँ पे डाई जो एंथ्राक्विनोन है, एंथ्राक्विनोन प्लस यहाँ पे ये हाइड्रोक्सिल रोटिकोनिक और यहाँ पे कॉमन रहेगा, यहाँ पे मिथाइल. स्ट्रक्चर आर नॉट मच इम्पोर्टेंट. देयर इज इमोडिन, एलोइमोडिन, फाइसिकोन एंड क्राइसोपेनॉल. So most of the this anthraquinone glycosides consist of these side chain or these A glycol part. Next one is Cascara. So Cascara is known as a Cascara sagrada. Cascara sagrada, sacred bark. Synonym से याद रखो, Cascara sagrada, sacred bark. Chittum bark and Prush blackthorn. Cortex ram prussiani. Biological and geographical source: dried bark of Rhamnus prussiana, family Rhamnaceae, found in the north. Okay, found in the North Carolina, Washington, and Canada and Kenya. So that is known as a Cascara sagrada. So again, it is again a laxative. Okay, laxative purgative. Now microscopic character just इसका important नहीं है microscopic character. Let's remember there is a prismatic calcium oxalate crystal. Chemical constituent, it has a C glycoside as well as O glycosides. Both type of glycosides are there. So known as a cascaroside A, B, C, D. Cascaroside A, cascaroside B, cascaroside C, and cascaroside D. So they are alpha configuration. Amongst these four, cascaroside D will have alpha configuration, and cascaroside A, B, C. These three have beta configuration. It also consists of some amount of barbelloin, chrysolene, dianthrone, imodine, aloimodine, and palmidine A, B, and C. So it is used as a bitter, stomachic, tonic, and purgative organ. So chemical test, there is nothing but both brown tracker as well as modified brown tracker. Substitute, that is nothing but this Rhamni californica, Rhamni felix, Prangula bark. That is used as a substitute. Not much important. Next, next drug is a hypericum. It is known as St. John wort. Hypericum or St. John wort. It is known as a hypericum or St. John wort. Saint John wort. Okay. St means Saint. Saint John wort. Melipertis and goatweed. Remember goatweed. बकरी का चारा. Hypericum. Okay. So biological source dried aerial part of hypericum purpuratum. Family Hyperaceae. Hyperaceae. Okay. Found in the Europe, Australia, Western Himalayas of India. Chemical constant क्या naphthodianthrone like hypericin is the glycoside. It is a meso compound and it has a red fluorescent pigment. That is nothing but the hypericin. Hypericin is a nothing but the naphthodianthrone. मतलब naphthacin रिंग होगा उसमें डबल डायएंथ्रोन नेप्थोसिन डायएंथ्रोन. जैसे एंथ्रासिन होता है 3 रिंग, नेप्थलीन होता है 2 रिंग. 2 rings of benzene plus ketone, naphtha dianthrone, and double that's dianthrone. Hypericin. It is a meso compound and it is a fluorescent pigment. Then pseudohypericin, hyperoside and flavonoid like amantoplavin. Uses it is used as a antidepressant. Is an antidepressant. It is used in the neuralgia, fibrosity. Neuralgia means nerve pain, menopausal neurosis, and it is also antiviral agent against the HIV and hepatitis C. So that is nothing but the hypericum. Now we'll see the aloe vera. Let me check once. Now this is aloe vera. You might have seen this plant lot of times. आपके गार्डन में भी होगा, कॉलेज के गार्डन में भी होगा, आपके घर पे भी होगा. बहुत सारे जो गर्ल्स है वो भी यूज करते हैं डेली इसको एज अ कॉस्मेटिक बोल के. उसका जो ये जो है whatever juice is present in this aloe vera that is used as a multiple application in the cosmetics also. So we'll see it. So aloe vera, you have different varieties of aloe. Okay, so the synonym for this aloe vera, so it is also known as aloe, then musabar kumari, aloe, musabar and kumari. So remember kumari means girl, and girls used to apply this juice of aloe vera on their cheeks, so that's why that is known as a kumari. And what is it? Biological source and geographical source. So dried juice of aloe barbadensis. Dried juice of aloe barbadensis is known as a curaco aloe. There are different varieties. So aloe barbadensis is known as a curaco aloe. Then aloe peri, that is known as a socotrine aloe. aloe peri का variety है तो उसको बोलते हैं socotrine aloe. aloe barbadensis है तो उसको बोलते हैं curaco aloe. Then aloe ferox का variety है that is known as a cape aloe. aloe ferox and other species aloe ferox का species है या फिर दूसरा कोई species unknown. If aloe barbadensis is there, that is known as curaco aloe. If aloe peri is there, that is known as socotrine aloe. If aloe ferox and other species of aloe, that is known as a cape aloe. All belongs to family Liliaceae. Now cultivation leaves are sessile and have strong spine at apex. Sessile मतलब So there is only one leaf, they are not present in the pairs and all that. एक ही लीव रहेगा, the sessile and have strong spine, उसको एक नोकीला अपेक्स रहता है, spines along with the margin उसका सीरिएट मार्जिन रहता है. रूट सकर्स आर यूज्ड फॉर द प्रोपोगेशन, मतलब उसका जो रूट होता है या फिर जो रूट के जैसा रूट का स्टीम होता है, दैट इज यूज्ड एज द रूट सकर इज यूज्ड फॉर द प्रोपोगेशन. वो लगाया आपने तो ये पूरा प्लांट ग्रो हो जाएगा. नाउ दिस प्लांट कैन ग्रो इन द पुअर सॉइल एंड ड्राई कंडीशन. It is cultivated medicinally. It is planted at a distance of 50 centimeter. Water logging should be prevented. जहाँ पे पानी का जमा होना वहाँ पे नहीं लगना. So water logging should be prevented. And for a good medicinal purpose, you have to use manures like your nitrogen, potassium, and NPK. Your manure, nitrogen, potassium, phosphorus. So that is used. The leaves are started to cut in the second year of the plantation. ये प्लांट लगा है आपने सेकंड 2 साल का हो गया तो उसके बाद में आप उसका निकल सकते हो फॉर मेडिसिनली. सो इफ यू आर कल्टिवेटिंग दिस आलो medicinally or medicinal cultivation of aloe, the leaves are cutted and collected second year of the plant, and it can grow up to 12 years. You can collect the leaves up to 12 years. Preparation of aloe. Preparation of aloe. Different uh, preparation of aloe vera juice for market. Okay, so depending upon the variety and depending upon the variety and preparation method, they are divided into different types. First one is Barbados or Curaçao aloe. इसमें क्या होता है कि whatever leaves are there, they are cut and kept into kerosene tin. जो आपका रॉकेल का कैन होता है उसमें डालने का, they are cut and kept into kerosene tin because of spine. The cut leaves are placed in the V-shaped. Cut leaves are placed in the V-shaped wooden tub. लकड़ी का टब बनाते हैं वी शेप्ड, उसमें वो सब जो कट किए वाले लीव्स रखते हैं सो दैट द जूस विल बी ड्रेन्ड ओवर द इन द टब. सो जूस इज इवापोरेटेड. Okay, then that juice whatever collected in the wooden tub, that is evaporated the copper pan then poured in the guards or metal to harden. Guards or metal to harden is उसका डाइज होते हैं, डाइज call them as dyes, guards or metal to solidify that juice. That is known as they are marketed under the name of Barbados or Curaçao. The next one is known as a Cape Aloe. Again, you have to you have to collect the leaves, cut and arrange in a circular manner in a basin shaped depression dug in the ground, lined by the goat skin or canvas. मतलब क्या करना है, आपके जमीन में गड्ढा खोदना है. ओके, गड्ढा खोदना है और उसके उसको गोट स्किन, जो बकरे का स्किन है वो उसका लाइनिंग, मतलब उसको बकरे का स्किन बिछा देना है उसके ऊपर. बाद में वहाँ पे रोटेट शेप में वो जो लीव्स है वो रखना है so that the juice will be collected in the goat skin. Okay, now that collect the juice and boil in the iron kettle, then stir continuously with a wooden paddle and then pour into wooden cases to solidify, and that solidified aloe is marketed under the name of Cape Aloe. Then again Socotrine aloe is there. Socotrine aloe. So again you have to collect the juice, the goat skin, and then you have to allow the to solidify. That is a Socotrine aloe. Then gingiber aloe, same as like above method, but the source of that aloe is unknown. So there are four varieties of aloe for the market. That is nothing but the Barbados or Curaçao, Cape aloe, Socotrine aloe, and gingiber aloe. अगर कभी क्वेश्चन नहीं आया, लेकिन फार्मासिस्ट एग्जाम जो होता है उसमें आ सकता है क्वेश्चन. Now we'll see the chemical constituent and all that. So, microscopic character. Okay, so microscopic character में एक point important है आपका यहाँ पे. Aloetic cells. Aloetic cells consist of a viscous yellow liquid or yellow juice known as aloe gel. Okay, so whatever the aloetic cells are present in the leaf of aloe vera, so that cell secretes and contain viscous yellow juice known as aloe vera juice. Okay, so again this aloe vera consists of calcium oxalate crystals which are present in the parenchymatic cells. Just remember the aloetic cells. So juice present in the aloe vera that is secreted by or that is stored in the aloetic cells. Okay, chemical constant. Now it is important again. Here anthraquinone glycosides are there. So main glycoside is nothing but the barbaloin. Main glycoside in the aloe vera is a barbaloin. Is a cheap, that is a maximum component or cheap constant of the aloe dianthron. तो barbaloin जो है that is the main anthraquinone glycoside और उसके उसका भी cheap constant मतलब so barbaloin is the cheap constant which is nothing but the aloe anthron that is 10 carbon glycoside that is it is a water soluble. This barbaloin is a water soluble and also consists of isobarbaloin and beta barbaloin. Aloe barbaloin is the main content which is having nothing but the aloe anthron C10 side chain. It is responsible for the purgative activity. So purgative activity of your aloe vera is because of this barbaloin. Curaco aloe consists of the highest amount of barbaloin. 2% barbaloin is present in the Curaco aloe. So variety में ज़्यादा आता है Curaco aloe. Curaco aloe will have a highest amount of barbaloin. Now barbaloin, isobarbaloin, beta barbaloin, they are the isomers of barbaloin, the isomers of aloe. Now along with this, there is one resin in this aloe vera known as aleucine. Aleucine is the resin responsible for the purgative action, and it is a its type of C-glucosyl chroman जो है laxative and purgative activity अगर पूछा है आपको तो barbaloin बोल दो. अगर the resin responsible for the purgative activity ऐसा पूछा है तो aleucine. The use of this, okay, it is a purgative ingredient of mixture of tincture benzyne. Tincture benzyne जो है that is a Farrar's blossom benzyne. उसका ये ingredient होता है. Then prepared thorax balsam of aloe. Farrar's balsam जो होता है या फिर tincture benzyne जो होता है that is prepared from storax, then balsam of then aloe and 90% alcohol. That is nothing but the Farrar's balsam benzyne or known as a tincture benzyne. Okay, it is used as a purgative also, and it is a constant of your Farrar's balsam benzyne. Then again in a cosmetic, it is okay in the cosmetic. It is used in the cosmetic as well as skincare product. It is a aloe vera juice is anti-inflammatory due to presence of salicylate. Salicylic acid derivative होता है इसमें aloe juice में. That's why it is a anti-inflammatory. It is used in the cosmetic as well as skincare product. Salicylic acid and carboxypeptide is present in the juice of aloe vera, has an exfoliating action. Exfoliating means removal of dead cell from your skin. Exfoliating action that is responsible for the removing the dead cell from your skin. That's why it is used in the cosmetics. Salicylic acid and carboxypeptide is enzyme in the aloe vera juice that is responsible for the exfoliating action. To stimulate the macrophage production means it has immunomodulatory action. It is used in the treatment of burns and wounds. जो भी जख्म है burns हो गया. Next one is your chemical test for aloe. Different, there are different varieties of aloe and there are different chemical tests. First you have to use general test. General test or you have to make a solution. Jam of powder 10 ml of water boil. Cascara filter, then bromine test you have to perform. These some tests are important. First one is the bromine test. Prepared bromine solution plus filtrate will give you yellow tetra bromelain. करना है 1 ग्राम ऑफ एलो पाउडर लेना है आपको 10 ml वाटर लेना है बॉईल करना है फिल्टर करना है उसको यूजिंग केसलकर एज अ filtrate. Now that filtrate is treated with the freshly prepared bromine solution, you will get a pale yellow precipitate due to tetrabromelain. Then again that filtrate is used for a second test, showing 10 tons reaction, or it is known as a borax test. Showing 10 tons reaction or borax test. So filtrate plus borax to dissolve, shake well, add few drop of solution into nearly filled nearly new test tube in which there is water, so it will give green fluorescence, that is known as a borax test. Different variety of aloe will give you different, example nitric acid test, sodium nitrate with acetic acid in presence of aqueous solution of aloe vera. Now curaco aloe will give you sharp pink color to coramine color, then cape aloe will give you a different color. These are not much important. Curacao aloe will give you pink color or caramel color. Cape aloe will give you pink or paint color. Ginger and Socotra aloe will give very less change in the color. Okay, nitrous acid test. Then there is another nitric acid test. Solution of drug, मतलब aqueous solution of aloe vera, aqueous solution of aloe plus nitric acid, aqueous solution of drug plus copper sulfate. Then you have to add sodium chloride in excess of 90% alcohol. This is the test procedure, which is mostly short में दिए हैं. Aloe will give you deep yellowish red color. Cape aloe brownish green color. Gingiber yellowish brown. Socotra and pale. ठीक है? कुछ ऐसे ये colors देंगे, which is not important. Then this test is important. Cupraloin test, or is also known as Clunge isobarbeloin test. This test is important. So in case of Curacao, wine red color is given. You get wine red color. Capello, like color is not much important. They will not ask you which color is gone by which variety. Just remember Cupraloin test is also known as Clunge isobarbeloin test. Then modified anthracnone test. Modified anthracnone test, like modified prawn tiger test की जैसे ही होता है. All they are going to ask you 11 question form. So now we'll see the cardiac glycoside. Now this component is important. Simple एक दोन वेळेस वाचलं ना तुमच्या लक्षात राहून जाईल. कारण आता aloe मध्ये काय काय आहे, aloin, allosine. So simple things आहे. Barbeloin is a nothing but the glycoside, anthracnone glycoside. Allosine is nothing but the resin responsible for the purgative activity. Barbeloin is responsible for the purgative activity. ठीक आहे, असं एकदम सिंपल असते. आता कुठल्याची केमिकल कॉन्स्टंट लक्षात राहत नाही सांगा मला. Senna मध्ये काय, सेनोसाइड A and सेनोसाइड B, सेनोसाइड C, सेनोसाइड D. So out of that सेनोसाइड B is responsible for the purgative activity. What is that? दूसरे भी इसमें क्या है, वही थिंकेबल. रूबार्ब में क्या है, that is nothing but the, ठीक है, रूबार्ब में क्या है आपका केमिकल कांस्टेंट. सिंपल थिंग्स है उसमें भी. एंथ्राक्नोन ग्लाइकोसाइड से सिंपल. बाकी ये परमेडिन A B C बस इतना याद रखो. बाकी उसका जो है एक्सटेंशन, जैसे अलोइमोडिन, सैंथ्रोन, डायंथ्रोन, ये छोड़ दो, उसका नहीं कुछ. Obviously, any glycoside containing any glycoside containing O glycoside ज्याच्यामध्ये ऑक्सिजन इन्व्हॉल्व आहे बॉन्ड फॉर्मेशन मध्ये, कुठल्याही प्लांट मध्ये जर असेल ना O ग्लायकोसाइड, तर ती टेस्ट पॉझिटिव्ह देणार. ब्रॉन ट्रॅगर टेस्ट. सो पावडर ड्रग प्लस TH2 सल्फर, hydrolyzed TH2 sulfur, then add, then extract with the organic solvent like chloroform ether or form ether or benzene, and then add ammonia. So you will get a pink color with the presence of anthracnone glycoside. That is a Brown Tracker test. यहाँ पे तुम्ही फक्त एवढं लक्षात ठेवा, Brown Tracker test is for O glycoside, modified Brown Tracker is for C glycoside. That is important. And aloe vera चा यूज काय आहे. सो इतकी सिंपल क्वेश्चन येतात ना GPAT वरती, ह्याच्यामध्ये जास्त काही विचारण्यासारखं नाही. विल सी द लिस्ट ऑफ